Hello classmates, today I'm gonna teach you about Introduction to Heredity. Our particular topic, Mendelism, Classical Genetics. Now let me introduce you to Gregor Johann Mendel. He's the one who discovered how traits pass from one generation to another. He is a scientist and or a botanist. He is also a monk based from what I have read. He is the first person to lay the foundation of the science of genetics. So just like what I have said earlier, I'll just give some example to you to explain this. Some example is eye color. So we have two eye color, the blue eyes and the brown eyes. So this is two different type of allele. So what is allele? Allele is a specific version of a gene. So for example, we go to its microbiology to understand more clearly. So for example, let's say on some chromosome, there are some location here. We call it locus. So where the eye gene is located. For example, from your father, the locus of the brown color is located here, and from your mother, the blue color is located here. So what is locus? Locus is a specific location of gene on a chromosome. So we have here the example. The brown color is the dominant. So we have here the word dominance. The brown is dominant. The represent the capital B because it is dominant. The blue is recessive, represented by small letter B, since it is recessive. So here we have heterozygous because it has two different alleles, the one recessive and one dominant allele. So, and we have here the two identical alleles, the two recessive gene, and it is called homozygous. What I have wrote here is genotype and phenotype. Genotype is the specific gene you have, while phenotype is what is expressed, what do you see. So just like what I've said, the capital B represent the dominant allele, brown eyes, and the small b represent the recessive allele, which is the blue eyes. And here we have example heterozygous because it has two different allele, dominant and recessive and dominant. So it will coded or it will have brown eyes because it has dominant. Okay. Here we have dominant allele. It will coded brown eyes. We have here the homodominant, two dominant allele, brown eyes and brown eyes. It will also coded brown eyes. We have another homo recessive from the word homo, which is the same, two recessive allele. It will coded blue eyes. So this is just a simplification on how heredity works. So in here you can see that there are different genotype. We have three different genotype, but it will coded to have the same phenotype: brown eyes, brown eyes, brown eyes. In this context, it shows also that there are different allele for the same gene. There are different allele, 1, 2, 3, but it will have the same gene: brown eyes, and brown eyes. You will have brown eyes, but having two different alleles. Now I'll give you another scenarios on what is really happening during reproduction using the Punnett Square. So Punnett Square helps us to track dominant and recessive allele pairings that make up a trait's genotype. We have here example, we have your parents gives off two different alleles, the dominant, recessive from your father, recessive and dominant. So the Punnett Square has 4 boxes. The first box has two dominant allele, one dominant from your mother and one dominant allele from your father. The second box has heterozygous, dominant allele from your father and recessive allele from your mother. The third box has again heterozygous, the dominant allele from your mother and the recessive allele from your father. The last box has homo-recessive, recessive allele from your father and recessive allele from your mother. From this Punnett Square, we can conclude that there are 75% or 3 out of 4 chances that their child will have brown eyes, and 25% or 1 out of 4 chances that their child will have blue eyes, and 50% or 2 out of 4 that their child will neither have blue or brown eyes, heterozygous. Now we go to another topic, the Mendelian inheritance, particularly autosomal recessive and autosomal dominant. This disease only occur and caused by single abnormal gene on a non-sex chromosomes. The single gene disease occur in non-sex chromosomes. These chromosomes are not X or Y chromosomes. So these non-sex chromosomes is also called autosomes. Autosomal recessive, autosomal dominant. There are 2 examples of diseases that are caused by single gene disorder that can be passed on. These are cystic fibrosis and Huntington's chorea. The differences between these 2 diseases is that Huntington's chorea gene is dominant, while cystic fibrosis gene is recessive. To have Huntington's chorea, you will only need 1 copy of the disease gene. Only need 1 disease gene, because it will overwrite or change your other gene, which is normal. When you have 1 disease gene, the disease gene will override the normal gene. The normal gene will also become the disease gene. While on the cystic fibrosis, you will need 2 disease gene, because if you only have 1 disease gene, the normal gene will overwrite the disease gene and it will have 2 normal gene. So when you have 1 gene of disease gene, you will be called carrier, because you don't actually have the disease, but you have the capacity to pass it on to your offspring. So now I'll be teaching you how to calculate the possibility or risk on one of the offspring having autosomal recessive condition. In order to calculate the risk, it is very helpful to draw an inheritance pathway. By using this method, you can always work out the risk of various outcomes based on the genetic status of the parents. So I'll be using some example to make it clearer. First, let's consider that both parents are carrier of autosomal recessive condition. Each parent has disease gene and a normal gene. As a result, they are both carrier. They don't actually have the disease, but they have the capability to pass it on to their children. I labeled the copies of the gene that could be inherited from the parents as copy 1 and copy 2 from the father, and copy 3 and copy 4 from the mother. There is always 4 possible outcomes to the inheritance of this particular gene: 1, 2, 3, and 4. Outcome 1 is copy 1 from the father and copy 3 from the mother. Outcome 2 is copy 1 from the father and copy 4 from the mother. Outcome 3 is copy 2 from the father and copy 3 from the mother. Last, outcome 4 is copy 2 from the father and copy 4 from the mother. Outcome 1 has 2 copies of the disease gene. Therefore, this person would have the disease. Outcome 2 has 1 copy that codes for the disease gene and one normal copy. Therefore, this person is carrier just like their parents. Now, outcome 3 has one copy of disease gene and one copy of normal gene. Therefore, there are they are carrier as well. Lastly, outcome 4 has two normal copy of the gene. Therefore, this person is free from the abnormal gene and does not have disease and not a carrier. Outcome 1 has 1 in 4 or 25% chance of having the disease. Outcome 2 and 3 has 2 in 4 or 50% chance of having or being a carrier. Outcome 4 has 1 in 4 or 25% chance of having normal genes. So by doing this, you can calculate quite easily the risk on children of being or having the disease, being a carrier or having normal genes. Now we go to another topic, the autosomal dominant inheritance. A quick review. A human has 46 chromosomes in every cell in their body. It consists of 22 pairs of chromosomes, numbered 1 to 22. So these 22 pairs of chromosomes are your non-sex chromosomes. That 23rd or last pair of chromosome is the sex chromosome. These 1 to 22 chromosomes are called autosomes. So these chromosomes, the pairs of these 22 pairs of chromosomes, are the same in males and females. These chromosomes carry about 22,000 genes. Autosomal dominant inheritance. So what is autosomal dominant inheritance? Autosomal dominant inheritance describes the situation wherein there is an altered gene on the 22 autosomes, and someone who carries this altered gene is affected by the disorder. Many autosomal dominant conditions can vary in their severity between one person and the next. Sometimes you can get variability between affected members of the same family. You can also get variability between unrelated individuals that has the same genetic condition. Sometimes you might find that both parents are healthy, but their child has an autosomal dominant condition. This is because a new mutation has occurred in the dominant gene in either sperm or egg. Examples of genetic conditions due to autosomal dominant inheritance include neurofibromatosis types 1 and 2, tuberous sclerosis, achondroplasia, and brittle bone disease. The question is, why is that the autosomal dominant alteration has more variability? It is because it has more severe effect on the way a gene works compared to other alterations. Sometimes an alteration can totally disrupt a gene, while other times the altered gene can still function in part. Now I'll show you how to calculate the risk of passing on an autosomal dominant condition. We will be using the same method that we did for autosomal recessive condition. In this example, one parent is affected by the disease and the other one is disease free. So again, we start with 4 copies, 2 from each parent. Let's assume that copy 1 is copy for the disease. Outcome 1 is copy 1 and copy 3. Outcome 2 is copy 1 and copy 4. Outcome 3 is copy 2 and copy 3. And outcome 4 is copy 2 and copy 4. Therefore, outcome 1 and outcome 2 has the disease and outcome 4 and outcome 3 has normal genes. We can conclude that children from these parents has 2 in 4 or 50% of having disease, and 2 in 4 or 50% chance of having normal genes. So I hope this explanation had made it a lot clearer, and I'll see you next time.